Hello friends, this is Akshay More. In our last lecture, we study about complementary genes. So today we are going to start our next topic, that is duplicate genes. 5 is to 1 ratio. Next topic be duplicate genes. Duplicate genes 15 is to 1 ratio. Okay. So friends, what is duplicate genes? When a dominant allele at either of two loci uh, can match the expression of recessive allele at the two loci is known as duplicate dominant epistatics. So, what is duplicate genes? The dominant allele at either of two locus or loci can match the expression. So, of the recessive allele at the two loci, it known as duplicate dominant epistatics. Friend, uh, we take an example, that is a rice character. Okay, in a rice, one character is controlled by two dominant duplicate genes, that is capital A and capital B. That is gene capital A and capital B. Okay, so the presence of any of these two alleles can produce one. So the oneless condition develop only uh, when both those genes are in homologous recessive state, that is uh, small a, small b and small b. So a cross between on and onless strain produce own plant in F1. So right here, these are the parents. First one I write here parent. Okay, first character, uh, capital A, capital A, small b, cross between small a, capital B and capital. This is the own rice right here, own rice, and this one the unless rice, unless rice. Okay, so I cross between those characters. F1 is capital A, small a, capital B and small b. That is called as own rice. Okay, own rice. So the selfing of F1 plant allele or F1 plant produce own and unless plant in 15:1 ratio in F2 generation. So we are going to draw a Punnett square, but the allele is epistatic to a small a or small b allele, and the all plants having allele a will develop on another dominant allele capital B. So the epistatic to allele small a and small b. So I draw here Punnett square for F2 generation. So what is F1? I write here F1 generation, capital A, capital B and small b. This is the our F1. So I draw Punnett square for F2 generation. Okay. So 1, 2, 3 and 4, 5. 1, 2, 3 and 4. Okay. So these are the male and this one the. Okay. Right here their gametes, that is capital A, capital B, capital A, small b, small a, capital B and small a, small b. Capital A, capital B, capital A, small b, capital A, small a, capital B, small a, capital B and small a, small b. Okay, so right here capital A, capital A, capital B, capital B. Okay. Uh, right here, capital A, capital A, capital B, small b. Capital A, small a, capital B, and small b. Okay. Capital A, small a, capital B, and small b. Capital A, capital A, capital B, and small b. Capital A, capital A, small b, small b. Capital A, small a, capital B, small b. Capital A, small a, small b, small b. Okay. Capital A, small a, capital B, capital B. Capital A, small a, capital B, small b. Sorry. Small a, capital B, capital B. Small a, capital B, small b. That's one. Capital A, small a, capital B, small b. Capital A, small a, small b, small b. Capital A, small a, capital B, small b, and this is the small a, small b. So this type of ratio we call as 15:1 ratio. 1, 2, 3, 4, 5, 6, 7, 8, 9, 12, 11, 12, 13, 14, and 15. So this ratio we call 15:1 ratio. So this type of gene we call as duplicate gene. Friends, hope you understand this topic. Thank you.